A very good morning to all, and so we are here. I welcome again to all the participants and to our speaker of the day. So today is the second day of the FDP. As you all know that this session is going to be truly international because both the speakers are from international universities. Today, before I introduce the first speaker, I would request Dr. Shreya Nair to briefly tell us about the guidelines because many of the participants are facing difficulties in sending the feedbacks and many other things. So Shreya Nair will explain us briefly the guidelines. Shreya Nair, please.
Yeah, good morning to all of you. I had received so many queries from the participants about how to access the program. So I will clear it now. So please follow these steps. Please go to the concerned speakers page in the website for each sessions. For example, if the session is day 2, time 2:30 to 3:30 p.m., then you have to click the speaker profile. For example, Professor Patricio's profile. You will be directed towards his page. There you should see a button to join the session, and after clicking the button, you will be directed towards a Google form and fill the details as there. And on submission, you will be seeing a link for the session, and click on that and join the session. When the session is over, please come back to the concerned speakers page on the website and fill the feedback form. Note, feedback link will be open till midnight on that particular day. And after that, you will not be able to access the feedback form, leading to the failure in completing this program. So please fill the feedback form at once the session is over. Please follow these steps to access further sessions of this program. And all the details about the program will be available in our website. So do visit our website for any queries. Each and every session in this program is recorded, and the recorded videos will be available in our website and in our YouTube channel, Listen, Learn, Grow. Listen, Learn, Grow. Thank you. Over to Dr. Rajiv.
Thank you, Shreya. That was very comprehensively explained. I think the participants will follow all your steps. Sorry for the interruption, Dr. Nayeem. Now, let me start with the brief introduction of today's speaker. We have a very versatile, young, good-looking, handsome gentleman today for the very first talk of the day. So that is The Tale of Laughing Gas, a voyage to Arabian Sea. So we are expecting something new and innovative from today's talk. So I'll briefly introduce you to the speaker today. So Dr. Nayeem Mullungal is a faculty member in Department of Biology and Environmental Sciences at Qatar University, Qatar. Dr. Nayeem is a faculty member of Marine Chemistry in the Department of Biological and Environmental Sciences at Qatar. He completed his PhD in Chemical Oceanography from the Department of Chemistry, University of Otago, New Zealand in 2017 with an international student scholarship. We congratulate you, sir, for completion of your degree with an international scholarship. He acquired MPhil degree in marine chemistry with first rank from the Department of Chemical Oceanography, CUSAC, in 2011. Soon after completing his MSc in hydrochemistry from the same department, his major research focus is on the oceanic nitrogen cycle using novel stable isotopic techniques. He has presented his research works in more than 10 international conferences in 6 different countries. He has secured various international awards and supports for his work from agencies such as AGU, SOLAS, IAGS, NZ, MSS, and Chinese Academy of Science. So I am getting a feeling that this guy is going to definitely do a very good work. He's an early starter and a very young guy. So I welcome again Dr. Nayeem for today's talk. I welcome you. I hand over to Dr. Nayeem now. Thank you, sir.
Good morning. Good morning, all. Thank you so much, Dr. Rajiv, for the nice introduction and nice words. I'm really honored to speak here today in this one-week faculty development program, which is actually really highly interdisciplinary as well, multidisciplinary as well, organized by a lot of prestigious Indian institutes together, create college, ICAR, PSGR Coimbatore, and Maharshi, Dayanand University, Haryana. Before I start, I would like to, once again, I would like to thank you, the organizers, especially Dr. Shreya, who is one of my friends, for inviting me here and having me here. I know we all are going through very difficult situations, and it's a very nice platform to share the knowledge. Actually, we are still keen to gain knowledge. So my talk is about the laughing gas and its role in the ocean science, and I mean in the ocean, current ocean scenario. The tale of laughing gas, a voyage to Arabian Sea. So it's a kind of related to global warming, climate change. Till chemistry, obviously it's highly linked to the biology. So as your conference is, my talk is also highly, multidisciplinary. So here we go today. So first of all, I will give you a short introduction on the nitrous oxide, the laughing gas, and then I will give you a more detailed information how I did my work, how I'm going through the research. And I'm sure like this will be, for many of you, this might be a new insight into the marine science. And then we go through a little bit through my results, and I will conclude. I don't want to talk a lot, so my talk will be most probably 40 to 45 minutes. So though that my title is it's about laughing gas, nitrous oxide is not at all a laughing matter. Actually, when an environmental scientist talk to you, so nitrous oxide, the laughing gas, I won't be using the term laughing gas a lot. Instead, I will be using nitrous oxide or N2. And this is a greenhouse gas. You all are well aware about that, like carbon dioxide and methane. And apart from its role as a greenhouse gas, it is a precursor of the ozone depletion in the stratosphere. A lot of research are going through in that side as well, and even there was scientists who got Nobel Prize for the studies related to the ozone depletion. So these are the two importance of nitrous oxide. One, it is a greenhouse gas, so obviously it's linked to the climate change in that aspect, and also it's a precursor of ozone depletion in the stratosphere. What makes N2O different from carbon dioxide or methane? We know carbon dioxide is more important when we talk about global warming and climate change. And when we talk about greenhouse gases, first comes the name of carbon dioxide. But if you look at, you can see when you consider 100-year, century-long effect, the potential of nitrous oxide, you can see here in my slide, it is around 300 times larger than carbon dioxide. And it got an atmospheric lifetime of 100+ years. And you will have a look at the graph here, NOAA data. You can see the nitrous oxide concentration in the last 20 years itself. It increased from 300 to 330. Now it's around 330 ppb. And now have a look at this one. This is a more, we all talk a lot about, environment, global warming, climate change, and most of us, irrespective of which field of science we are from, we are aware about this graph. Have a look at this one. Look at carbon dioxide, methane, and nitrous oxide. And if you look at not only nitrous oxide, if you look at any one of them, you can see that their concentration in the last 225 or 250 years, until then it was a very stable level in the atmosphere for long years. But now it's highly increasing. There are climate deniers. I'm not gonna talk about I'm not gonna debate on that, but this is the fact in front of us. And when it comes to nitrous oxide, and you can see in the last 250 years, as compared to the pre-industrial period, the concentration is highly increased. And more importantly, in future, a more increase of nitrous oxide is suggested. Why? Because of the increasing suboxic regions or anoxic regions in the coastal systems. That's very obvious, and a lot of scientists suggest it's gonna be more anoxic and suboxic zones coming in the coastal regions of the whole Arabian Sea, oxygen minimum zones. Plus the eutrophications and increase in productivity, a lot of other factors gonna add the N2O concentration more and more in the coming years. But the problem, as compared to, still our climate, global warming and climate research is more on carbon dioxide and methane than nitrous oxide. So as compared to the first two, especially as compared to carbon dioxide, the N2O data is still less, or we have a paucity of data. You can see, this is a screenshot from one of the recent research, which suggests this is the reason, nitrous oxide is even higher, or its role is even higher than that we know until now. So it's one of the highly underestimated parameter in the ocean. Now, the N2O, it's not only from this one, you can understand the different sources of nitrous oxide. See here, you can see in the circle, around 31% is from the ocean, including coastal regions and estuaries. And if you look at this one, you can see a lot of them are anthropogenic sources. Among the natural sources, ocean is one of the big contributor, or the aquatic systems are the big contributors. So how nitrous oxide is formed in the ocean? The way it is highly related to the biology as well. It's fully biological process, the conversion of nitrogen to the bioavailable nitrogen forms. And you all are aware, I hope you all are aware about the nitrogen cycle, which takes place in the ocean. So it's mainly driven by the microorganisms and the major pathways that leads to the formation are nitrification and denitrification. And in the last 10 years, there are studies, new insights, which suggest there is a lot of contribution from archaeal production as well. So here is the oceanic nitrogen cycle. If any one of you are not very well aware, or just want to recap the memory, I have a modified figure here which shows you in the left side the nitrification, how the nitrogen from the atmosphere, when enters to the ocean, first fixed into the ammonia and then finally converted into nitrate. And the opposite of this, which brings back the nitrogen back to the atmosphere, which is denitrification in the right side. And you can see the N2O or in the middle and its pathways. You can see this N2O is formed both from left side and right side. That means both from nitrification and denitrification. And for this figure, I will be using again later when I talk about isotopes. So this figure shows you 3 different pathways how N2O is formed from nitrification. See here, when ammonia is converted to NO3, then ammonia NH3 to NO3. In the middle, in the middle of the process, you can see a lot of intermediate products like NH2OH, which is hydroxylamine, NO, nitric oxide, NO2, then only it converted to nitrate. But you need to know that, or we need to understand that this N2O is formed from hydroxylamine, from NO, from NO2. This depends, you know, how it is formed from NH2OH or how it is formed from NO. It depends upon the different, you know, biogeochemistry of the ocean. And so again, this is the another figure showing the formation of N2O, so which we already have been through. Here I just want to focus on this part. Nitrous oxide production is highly linked to oxygen concentration in the ocean. When the oxygen concentration go less than the, you know, ambient conditions or the optimum level, that triggers the production of N2O. So now we have an idea how N2O is formed, from where it is formed, why ocean is, how much ocean contributes, how it is formed in the ocean. And now have a look at this one, different parts of the ocean, like, you know, environments like estuaries, coastal upwelling zones, continental shelves, and open oceans. Have a look at this. I have the first column here, second column, which tells you the percentage composition of the estuaries to the total ocean. See, only 0.4% is estuaries when you consider all of them in the total oceanic era, oceanic environment, while they contribute around 10% to the total nitrous oxide. So that's a very important thing. Why estuaries are contributing this much? Why coastal upwelling zones, which is only 0.2%, contribute 35% to the total nitrous oxide concentration? I hope you all can hear me, right?
Yes, you are audible, sir. Please carry on.
Right. So that's another thing. Continental shelves or open oceans, and at the same time, open oceans, which occupies around 82% of the world oceans, contributes only 40% only. So we are going through these things in a few minutes anyway. So what are the factors that controls the nitrous oxide production? Mainly the concentration is controlled, whether it is denitrification. We know it's these processes: denitrification, nitrification, and archaeal production. These productions and these biological processes with the microbes are highly controlled by the oxygen concentration. The first and most important factor is oxygen. Then what else other than this? Only oxygen? No, there are many other factors. Anthropogenic inputs are there. That triggers the concentration of the nutrients. There are other factors like wind. There are other factors like the upwelling. That the upwelling and downwelling is the oceanic feature, which brings highly nutrient and nutrient-rich water from the bottom of the ocean to the surface of the ocean, which takes place even in our nearest Arabian Sea. So when we go to the research side, what are the arguments and the questions? When I start my research, or even now, there is a debate actually on many things in this field. This nitrous oxide, when we go to, especially, we cannot go and take samples from the whole world oceans. It's a very, very highly collaborative nature work. Even after hundreds of scientists working together, we don't have enough data still on this question. But still, if you look at the coast of the Indian coast, and if you compare this to the New Zealand coast, I say the word New Zealand because my other research area was Pacific Ocean, and I was doing my PhD in New Zealand. So if you look at the New Zealand coast, the Indian coast is contributing much more the N2O source in the ocean. So in general, when we consider the whole world ocean, there are regions which are very less as compared to some other regions. So when you consider the global concentration of nitrous oxide, there are some regions in Southern Ocean, for example, which are like sink of N2O, while there are some other regions like that of our Arabian Sea or of Mauritius or of Chile. There are regions which are highly sourced to the global budget of nitrous oxide. So our question is that first one, the study regions are sink or source, first of all. Then we don't have even good idea about the distribution of this particular gas in the ocean. So how it is distributed in the water column from surface to the bottom. And then the next one, as I said, since dissolved oxygen is one of the very important parameter, how it varies with the dissolved oxygen, as the concentration of dissolved oxygen varies, how it varies. And there is a very, very important debate. That's the most important debate on the nitrous oxide ocean science, or which is the process of formation of N2O is from nitrification or denitrification, or there is another different process. There are a lot of scientists who debate for nitrification, while there are some other scientists who debate for denitrification. So the main objective of my study was to use some novel techniques of stable isotopes to look into this dilemma of nitrification or denitrification, or coupled nitrifier denitrification. So why Arabian Sea in particular? Because we have very nice scientists, a group of good scientists in India who worked a lot on nitrous oxide intensively for the last maybe 20-30 years, especially from the National Institute of Oceanography, Goa, and also from the Visakhapatnam. So when I was doing my research in the Pacific Ocean, I was interested to study our sea again, because there was not much study looking into the stable isotope aspect of the nitrous oxide in the Arabian Sea. So it was actually a new study. Plus, this region is already studied, so it will give a good comparative analysis to our data. So though Arabian Sea is only 0.43, less than 0.5% of the world oceans, it is contributing a lot to the global nitrous oxide flux. As you can see, it's around 35% to the upper level. So the reasons, some of the reasons, like the strong wind stress, and as I told you, the coexisting strong upwelling regions, both coastal and open ocean upwelling, and sometimes there is downwellings as well. And these are the major reasons for having a very high concentration in the nitrous oxide. But as I told you, there are very few studies on the nitrous oxide stable isotopes in the Arabian Sea, especially a very novel technique called isotopomeric studies, which is already done by a lot of eminent scientists from the United States and UK in Pacific and Atlantic, but we don't have that results from Arabian Sea in detail. So until now, before I started my research in the Arabian Sea, what we had, we did estimate the concentration of the nitrous oxide in the water. We from that concentration estimations, we calculated the flux and desired, when we estimated whether Arabian Sea is a flux or so, it's a source or sink for N2O. But our more studies are based on modeling works, but we don't have much results from the isotope related works. And there is no much works which is focused on the biological methodology as well. So before I go ahead now, I just give you a very quick overview. Might be because our talk is more into life science, I don't want to talk a lot into the chemistry, especially stable isotope chemistry. I just give you a few feedbacks, like what is stable isotopes. For example, we are aware about isotopes. When I talk about nitrous oxide, I need to focus on nitrogen and oxygen are the two elements present in the N2O. So nitrogen got mainly two stable isotopes, N14 and N15. The major concept here in the stable isotopes, in the natural environment, if there is no other threat or no other physical or biological processes that cause changes to one gas or one species or a molecule, the ratio will be, for example, N15 to N14 ratio is a constant value. So you took some samples from India, then I took some samples from Middle East, then I took some other samples from maybe Africa or Europe, and we estimate the ratio of N15 to N14 in the air or in the water. In general scenario, that should be almost constant. If it shows a very big difference, a huge difference, that means there is something interesting going on in that region where you have a very different result. So that's the key concept of our study as well. The similar to in nitrogen, oxygen got mainly three stable isotopes, 16, 17, and 18, and their relative abundance is as I have shown here, but the most abundant one is O16. So the ratio of the most abundant will be always considered. And how do I measure? It's called an instrument. The instrument is called IRMS, isotope ratio mass spectrometry or mass spectrometer. The instrument is called mass spectrometer. So it's mass spectrometry technique, and which is very specialized for isotope measurements that we have used in our studies. And you know, this is about the detector that we use in the equipment. More than the first one, the second one, the results are always reported as delta values, and in case of I mean relative to the air nitrogen, or and the results will be per mil, or you can see here the denotation for that. So you can see here, this is the way I calculate this is the equations that we use. I think like it will be more boring when I go to this part of this kind of explanations, but if any one of you are interested, just have a look at these ones, how I calculate N15 or how I calculate the O18. So this is the way I do it. First I calculate, I should have a standard, I should have a sample, then I just use these equations and I deduce the value of N15 and O18 for the samples. So in case of nitrous oxide, nitrous oxide is, as I told you, it got two stable isotope, the stable isotope of nitrogen and then that of oxygen. So why it is helpful? How the stable isotopes of nitrogen helpful in the in deciphering the biology of the nitrous oxide production? That's what I'm gonna explain in the next few slides. Because, you know, as I told you, sorry. See, as I told you, there is a mean value for N15 in the air, very close to 7.5 or close to 7. And that of, you can see, and that of oxygen is close to 44.6 plus or minus 0.2, and that means close to 45. This is the mean value of N15 of nitrous oxide. So you took air sample from Texas. You took overlying air sample from Haryana. You took air samples from Doha, where I am. Okay, 3 samples. You measure the N15 of nitrous oxide in that air samples. That should be very close to this value. If Haryana values are, let's say, 9.5, Texas values are 6.78, Doha values are 7.2. What does that mean? Doha and Texas values are close to the standard air value or mean air value, while our Haryana samples are a little bit high. Something interesting is going on. That's the key concept. So that's one thing. Another thing, how this nitrous oxide is coming to the ocean. Obviously, first nitrogen or N2O is coming from the air through mixing and wind kind of with the help of wind, mixing kind of processes takes place, diffusion takes place. So in general, by theory, if there is no other process, if the N2O formed, the N2O in the ocean is simply from the air, the N2O, N15 and O18 values in the oceanic water also should be close to that of air. So similar to 6.72 or 44.62, or it can be a little bit higher or lower. But if it's too much higher, like 9.2 or 10 or 4.2 or 2, that means there is some problem. Not problem, there is some other processes that either convert or takes nitrous oxide or produce nitrous oxide. So that is the major story or major information we get from N2O, but it's very pristine result actually. So that's the importance of that results. So that's what I have explained in the next slide. You can see here the reason, see, when by microorganisms convert ammonia or other things to a nitrate or a nitrite, what happens? Microorganisms prefer light isotopes than the heavier isotopes. So for example, let's say ammonia oxidizing bacteria. It goes and convert ammonia to, or oxidize ammonia. What it will do, it will take N14. It won't take N15. It will take O16. It won't, it don't like O17 or O18. So what will happen? It's a chemical process. So in the substrate, what will be left? OA17 or O18, right? And in the in case of nitrogen, it will be N15 left. So it will become, the substrate will become enriched in nitrate. I mean, in terms of nitrogen isotopes. And the reason for this, the molecules with heavier isotopes will react very slowly. That's the reason microbes prefer the lighter ones. So this is the way it, so as I told you, if there is a change in the, look at here, N15 values or O18 values than the normal value, than the mean value, that tells you some story about the process. Come on, there is something happening. That's why these values are less. So to get more insight, I also studied, oxygen isotopes. Apart from the oxygen of N2O, there are two other oxygen in the water when you consider this study. The H2O oxygen plus the dissolved oxygen, the oxygen that fishes breathe, that's the dissolved oxygen. Without that oxygen, fishes will die. So that's dissolved oxygen. That dissolved oxygen is not the oxygen of H2O. That's a very, whenever I give this talk, when you don't have a much idea in the marine sense, you might have a confusion. That's why I say this. The oxygen that fishes get in the water, dissolved oxygen, is different from the O of H2O. So what I did here, I studied oxygen, stable isotope O18 or of oxygen in N2O, in the dissolved oxygen O2, plus in H2O. Three things I have studied. And as I told you earlier, I want this figure later. I told you, this is the reason why I told you. See here, when ammonia is converted to hydroxylamine, the dissolved oxygen is added. And this oxygen in hydroxylamine O, that comes from dissolved oxygen. And if N2O is formed from pathway 1 directly from hydroxylamine, that oxygen is only the oxygen coming from dissolved oxygen or the oxygen that fishes breathe, right? But if N2O is produced through pathway 2, that is from NO, then again, it is same O. but there is a problem. If it is from pathway 3, that oxygen is a combination of already added oxygen from dissolved oxygen plus the oxygen coming from the water. So when you have two different oxygens that contribute to same species, the isotopic signal will be different. How it will be different? It will be sum of these two. So for, let's say, the oxygen delta value or delta, or the isotope signature for, normally we don't say oxygen value, we say isotopic signature. So isotopic signature of dissolved oxygen, let's say 20, and isotopic signature of H2O, let's say 0. Then this N2O, the third one, might have an isotopic signal close to 10, while this two will have a signal close to 20 itself. That's the difference. So with the values of oxygen isotopes and with the values of nitrogen isotopes, you are now very strong to say how the nitrous oxide is formed. And the third important parameter I measured is isotopomer. We need very highly sophisticated equipments actually to measure isotopomers. So isotopomers are actually one of a set of molecules having what isotopically substituted atoms. Look at this, these 5 species of nitrous oxide. All of them are nitrous oxide. Where they differs in the position of in their isotopes. Here, the first one got 2 of the nitrogen in the N2O, that belongs to N14. While in the second one nitrogen is N14, and while the other one is N15. So, so these are called isotopomers. Since another thing, the shape of the N2O, it's an asymmetric molecule. It does not have a symmetry, but it's a linear molecule. And since it's an asymmetric molecule, and if you measure this distribution, you can calculate, more. You can get a more good idea of the process again. Especially, it's called site preference. So if I call this nitrogen, the first nitrogen which is near to oxygen, as N alpha, and the next one which is far from the oxygen as N beta, and if I know the delta N alpha and delta N beta, what is delta? Isotopic signature. If I know the isotopic signature of alpha and beta in separate, then I can calculate something called SP. Next slide. Site preference. What is site preference? It is the it is just calculated by deducing the isotopic signature of alpha position, beta position from the alpha position. What is the significance of this value? It's a very good clear-cut picture. Is it close to 0? That means some process. It is close to 20. That means the process is different. So if you have SP values, no doubt, then you can completely say this. You are full 100%. You are sure of how N2O is formed. So, but the problem getting these results and getting samples ready and the instrument ready to analyze this is a little difficult. So I did a very detailed overview because it's a very different topic. Many of you might be not aware of. So I'm not going to talk about anyway my research in the other part of the ocean. I'm just focusing in the Arabian Sea. So I did, I did my study in these 3 regions, which is very close to the southern India. One is very close to the Cape Kanyakumari, and you can see the 3 stations in 3 different colors, and they're very much close to the coastal regions. And the importance of this region, I did sampling in monsoon, so they had a very high upwelling characteristics, but there was one station which is without upwelling as well. So this is the way I collect the sample. We use some special equipments called CTD bottles. From the CTD bottles, as you can see here, we draw the sample before we take any other samples for dissolved oxygen, for nutrients, for any other chlorophyll, any kind of analysis, first I collect my samples. This is special bottles called serum vials, which is around 250 ml capacity, and this is a tube called Tygon tubing. With using this tube, I draw the samples. I don't take, first of all, I need to put the tube inside the bottle. I need to be very careful that no contamination, no air bubbles. So sampling is a bit tricky. Then, this is the setup that I used in my lab. Actually, this is a reconstruction. This is a scientist from US, Casciotti. She developed a instrumental setup like this. When I started my PhD in 2011, my first task was to rebuild this one. So here it is. It took around 1 year for me to build this experimental setup. So you can see whatever I have here in this diagram is here as well. So here, I will quickly give you an overview. Here is the these ones. I don't have a pointer here too. I don't know. I can use a pointer or not. But anyway, you can see the sample rack here, where I put.
Yeah, the cursor is visible. The cursor is visible to us.
So you can see the sample rack, where I kept, once I finish my sampling, I can keep it for maximum 6 months. If I don't do analysis within 6 months, then there is a chance for contamination. So normally I do analysis within 2-3 days. I don't keep for long days, soon after my sampling trips. And you can see here 3 bottles already in the rack. What you can see a syringe. There will be a syringe here which comes to here once I give my start my program. Then the sample will come to this setup. Finally, there is a GC column here. There is a liquid nitrogen trap which is in blue, and you can see the traps. There's a lot of tubes there you can see. Finally, it will go down where is the isotope detector is, which is a mass detector. And I will go to my results. After this, I get results. I use some standards for nitrous oxides, which are distributed from some other international labs. And I also prepare some standard. If anyone of you are interested for the sampling technique, you can contact me, communicate me later. I think that's better about the standard preparation and related stuff. So my results are here for the, I'm not going to show all my results. I'm just going to show you to have an idea. See here, 3 stations I had, and 1 station was very shallow station, only 40-meter depth. So from that station, you can see in red, and the other 2 are the deep stations having depth around 1 kilometer. You can see this all from the deep station, you can see they are showing a single peak structure, 1 peak only. First, it is a little lower at the surface. When it reaches to the middle of the column or subsurface, it becomes very high. Then it reduces. So this is the normal result that is observed in the region from the previous works as well. But if you go to north of this region, up to Goan coast, the result will be a little different, because they have there in the northern Arabian Sea, there is a different biogeochemistry in the water column, with some regions having more denitrification zones. So there you observe a double peak structure, but in the southern tip of India, or in this southern part of the Arabian Sea, and in the southeast part of the Arabian Sea, you observe a single peak structure normally. But the thing here, you see here, in almost all stations, the surface concentration is above 10 nanomoles per liter, which is very high actually. Normally, the ambient air concentration is 7.2, 7.8, in that range when you say nanomoles. That is the concentration I received in the Pacific Ocean. While when you look at in the Arabian Sea, it is around 10, which is around 150 to 200 sometimes look at the UC station UC, which is a highly upwelling station. The concentration is close to 20, which is 3 times that of Pacific values. So that's interesting, actually, which actually going to prove that our region of the Arabian Sea is a highly important source of nitrous oxide to the ocean, to the atmosphere. But you can see here, so that's what I explained now, the distribution as here in the slide. If you compare this with the dissolved oxygen concentration, you can see in this part of the ocean, as compared to the surface at the bottom of the ocean, the dissolved oxygen concentration is very low. So if you compare this with this, you can see there is an opposite relationship. Normally, all researchers suggest this opposite or inverse relationship between N2O and oxygen is a sign of nitrification. So this is a preliminary. Earlier, maybe in 1990s, a scientist directly reached into a conclusion: the formation of N2O is from nitrification when there is an inverse relationship between oxygen and N2O. But nowadays, we cannot say it's only because of that. Sometimes there are caveats against this hypothesis. That's why we look further to confirm whether it is from nitrification or denitrification by using stable isotopes. So so that's the first story, a very preliminary result before I go to isotope results. But before that, I just want to tell you how, again, in detailed way, our part of the ocean in the Arabian Sea, is it a source of zinc? Look at this. This is the percentage saturation. So normally we calculate percentage saturation by comparing to the air equilibrium values of N2O. From this, when I calculated percentage saturation, have a look here, almost from surface to the bottom, the whole ocean is super saturated. Sometimes the super saturation reaches up to 600 to 700%. That means our ocean is super saturated, and it's a very high contributor. As I told you earlier, this is this proves why Arabian Sea contributing, though it is 0.4% of the total world ocean, why this contributes 30 to 20 to 30% of the total flux of the N2O. So I have also, by using some equations, I have not only one equation, I have used 3 different equations suggested by physical oceanographers to calculate the annual emission. So I have calculated from each stations, and then after that, you can see this is the nitrous oxide emissions in teragrams per year. And then there are other values calculated by scientists from the whole world ocean. And when I compare, for example, this per year estimation to the total world or global N2O flux from the whole world oceans, then I can say what is the percentage contribution to the total flux from the Arabian Sea. So this is the way I say this is total 35% of the world emission. Another way to look at the process for the production is having a look at the relationship between delta N2O and apparent oxygen utilization. If there is a positive correlation between N2O and apparent oxygen utilization, and also between N2O and nutrient, especially the nitrate and nitrite, that is also an evidence for N2O formation through the nitrification. But in our result, you can see it's a positive relationship, but they are grouped into 2. This means actually these results in the graph are actually one from the surface region, one from the bottom region. So bottom regions are grouped into 1, and the surface regions are grouped into 1. And in the mixing region, you can see they are spread over. So this proves it's not only nitrification. It's a clue that it's not only nitrification, but there is something else also going on. So we look into it, what it is. There is the role of isotopes. So here is my delta N15 values. you can see this. I told you earlier, the air values are close to 6.7. But look at here in the surface 0, the values are, except in some regions, the most of the values in the surface are less than the air values. Why? That means there are some biological processes that either reduce or produce N2O. So normally, if it is slightly lower, it's again a proof that it is produced through nitrification. Why? If it is denitrification, it is suggested that there will be highly enrichments. Instead of 7 point or 6 point or 5 point, if it is denitrification, it will be close to either 10, 10.2 like that. Or if there is denitrification which also consumes nitrous oxide, this may be negative. There are two possibilities with denitrification: either highly enriched values or highly depleted values. If it is a little bit depleted, then that means it's from nitrification. And you can see OH values follow same trend here in the next figure. OH values also follow same trend as in N15 values. So as I told you, the depletions in N2O are and enrichment in O18 a little bit at the bottom. It's due to the, especially here, there is an enrichment. If you look at here, in this figure, about 250 meter, you can see a little bit enrichment in O18. That enrichment plus in this figure, you can see a depletion at the same, about 250 in N15. I'm just repeating, so for those who are a little confused. In N15, in this figure, about 250 meter, you can see there is a depletion. While in O18, at the same depth, there is a enrichment. Enrichment means increase, depletion means decrease. So this contrasting values for N15 and O18, then that's another reason. It's not nitrification, it's not denitrification. That means there is another third process called nitrifier denitrification. So based on this, and also as I told you, I looked into it in another way. I looked into it by isotopomer studies also, as I told you. So I calculated the site preference values, and my site preference values are here. You can see it's not close to 0, except in almost all regions. It is, except in the one in the SK, the violet one, you can see in the first 2 stations, most of them are at the surface between 10 and 20. Yes.
You have pinned your. We can't see your slides, though we see your slides there in the smaller screen. But I think you should unpin yourself so that the slides can be enlarged on the screen. I think you have pinned yourself.
No, I don't do anything. Is it from the beginning same?
No. It happened just a minute before.
I don't know.
We see your picture enlarged, but your slide pictures are in a shorter screen.
Rajiv, this happened because some participant shared their screen.
Okay.
Is it still the same? Because I don't do anything. I just continue what I did from the beginning.
Have you pinned yourself? Because once you unpin yourself, so
no, I don't, because I didn't touch anywhere. I'm just continuing my talk. So as Dr. Sri has suggested, it might be someone else by knowingly or unknowingly did it.
Yeah. So one thing I suggest to the participant, they can pin this slide. They can see the slides in a shorter box. They can pin that to the screen. So once you pin it to the screen, you can see an enlarged version of the slide.
All right, thank you. That's a nice thing. So those who are having any issue with the slide, just follow what Dr. suggested. So the last one, okay. So the last one is about isotopomer, because I'm not focusing each of them. And if I want to get you, if I want to give you a good overview, I think I may need around 10 minutes for each on isotopomer, N15, O18, but because of the limitations, I'm just quickly going. But you need to know, I looked at the concentration, I calculated the flux, I compared this with the global value, then I calculated the N15, I calculated O18, then I reached into a conclusion it's mainly because of nitrification, the formation. But at subsurface, about 250 meter, there is a different process, which is nitrified denitrification, which I wanna, which all these my preliminary or my conclusions, I'm gonna make it more valid with the use of site preference values. And you can see here my site preference values also close to 250 are either very high, above 20, or very close to 0. This means at this particular depth, there is denitrification plus nitrification, coupled nitrified denitrification. Except all regions, the values are close to what we expect for nitrification. But if you look at here again, at the bottom, there is a slight opposite trend or there is a trend which is showing a little bit different process. That might be sediment denitrification. And apart from all this, I did one more study, which is called labeled isotope study. What is that? I use N15 and N15 labeled potassium nitrate, KNO3, and ammonium chloride. I collected samples, kept one sample as control without adding anything. To another second set, I added potassium nitrate. Another third sample, I added ammonium chloride. But remember, this ammonium chloride is labeled N15 and the potassium nitrate is labeled N15. So what is the advantage? If nitrification takes place, what will happen? What will happen? Ammonium chloride will be reduced. If denitrification is taking place, nitrate will be taken. So that's the, it's a very, highly, it's a very nice result if we get good result. So in my result, I found that there was no decrease in the concentration of N15 nitrate, while there was a clear evidence for uptake of ammonia or N15 labeled ammonium chloride. So I had a very convincing results for ammonium chloride, while there was not much relevant results obtained for nitrate. And this again proves the Arabian Sea region where I did my studies are producing nitrous oxide through nitrification. So what I concluded from my results: first, the area under study is an important source of nitrous oxide into the atmosphere, and the isotopic studies suggest that N2O is mainly formed through nitrification, with a slight contribution at the subsurface waters from nitrified denitrification. And that's all. And I would like to acknowledge all my colleagues and next to you people for your questions. But the most important stuff in the future for this study is biological. Now I want to do microbial studies. And I want to make sure that it's more. And if I want to make my results more excellent, I can do culture studies and DNA extractions, and I can reach into conclusion whether it is archaeal, whether is there any archaeal contributions, or is it nitrification is sure, or if it is, then what kind of organisms are doing nitrification. And if I can come up with that, it might be a very pristine result apart from this. So that's all, and I would like to thank you all again for your patience and your presence here. And once again, I would like to thank the organizers for having me here.
Thank you, Nayeem. That was indeed a new subject for us. We were happy to learn that the importance of the laughing gas. We thought that it was just a laughing gas. But today, you actually told us that it's really a big concern. And this is many times more devastating than the normal greenhouse gases like
Yeah.
CO2. So this is really interesting to learn, and your study was really. I think you should share your results with the government of India also. Uh, that this is a big concern, and since I'm not from this chemistry background, but I did appreciate your work and the way you carried out different experiments, the new setups you designed. That was really good to see that. So now I invite any questions from the participants. The session is open for any questions and answers. Naeem would be happy to answer. You can
There are,
write the questions in the chat box
yeah, in the
also.
chat box, there are two questions in the chat box, Rajiv, from one Dr. Sujata. Please.
Yes, please.
So what I can see in the chat box is appreciations, and people do
Is the
they
night
accept that
also
this
Ah,
this was new to them, and this was something which they learned from
I
you
can
only.
share my email address here for any further communication.
Right. Yes, we can see your email ID.
You
So
want my
many of our
email?
participants have appreciated your talk, and I see
Thank you.
one question. Yeah, one question is from Sujata. She says that how nitrous oxide is useful for the living organisms.
Actually, nitrous oxide is using your method can variables can be measured. So the first of all, how N2O is useful for the living organisms? You mean like how the organisms that live in the ocean, or the organisms that live in the can you make it a little bit clear?
Yeah, I think it would be good if you separately answer both, like for the living organisms in the oceans and then on the earth.
On the earth, actually nitrous oxide is as a greenhouse gas. We are talking as a greenhouse gas when it comes to the greenhouse. So I told you it's like carbon dioxide. It is trapping in the earth, like carbon dioxide. We know greenhouse effect. Carbon dioxide, what it is doing, it is trapping the heat. The problem now, the concentration of carbon dioxide is increasing, so it's trapping more heat, it's causing the global warming. Similarly, N2O is also doing the same job, but its concentration as compared to carbon dioxide is very, very low. It's in nanomoles, while carbon dioxide is in ppm. And another thing, but the problem N2O is 300 times when you consider in molecular basis. One molecule of carbon dioxide, you take one molecule of N2O. N2O can heat 300 times than carbon dioxide. We are safe, and we are, it's good that we don't have much concentration of N2O even now, but if it keep increasing, the effect of greenhouse or the greenhouse effect will increase more. But how it is useful? Yes, it is useful as a greenhouse gas, but carbon dioxide is also useful as a greenhouse gas, but it has other roles related to the plants. But nitrous oxide is not having any other role other than this to the plants or humans here in living in the earth. In the ocean, yes, it is a byproduct of this nitrification and denitrification process. So obviously, to go ahead with this process, or to have the nitrogen cycle going, it is very important to have N2O. If that process stops, all the organisms will be affected. As how the nitrogen cycle will be, how if anything happens wrong to nitrogen cycle, that will that will affect the earth and the environment. Similar way, it will affect the whole. It's a threat to all. If there is any very drastic increase or drastic decrease, it is very fatal to the whole. For whole species. For example, I would say like There is say like maybe 10 years back, there was a big experiment, to study the ocean. Like a group of scientists put a lot of iron to the ocean to see how it triggers the carbon dioxide concentration. It was done in both Atlantic and Pacific. A group of scientists, after their study, they found that when the carbon dioxide effect was, when the iron effect was increasing carbon dioxide, same time N2O was decreasing that effect. So, N2O has a big role as it is a part of nitrogen cycle. What will happen to nitrogen? What will happen to us if nitrogen cycle stops in the ocean for 1 day? It will be a big disaster to the whole world, actually, in a long term. So, yes, it's a very important thing.
Yeah, we have another question from
another one.
Dimpi Das.
Using
She
your
says,
before Dimbi Das, there is another one. Using your method, can biological samples be measured? Yes, I mean any kind of water samples or air samples, irrespective of it is biological or chemical, it can be measured. Yes. So I did not explain a lot about its working principle. Actually, when I share with you my equipment, what's doing there, the sample, you have seen there is a syringe that can go to the bottle. And what happens, that syringe will purge whole nitrous oxide, not only nitrous oxide, the whole dissolved gases in that bottle in around 40 minutes to a glass vial. Then it will be only gas. From that gas, I have a lot of different traps, which traps carbon dioxide, nitrogen, oxygen, etc. Finally, what I get in my liquid nitrogen trap will be N2O alone. And then I can just, with another set of airflow of free nitrogen or hydrogen, I can send my N2O to the sample. So if it is biological sample, I want to know what kind of biological sample. Then I may have to, I may be able to tell you what kind of treatments you need to do before you start your analysis. If in future nitrous oxide sequestration is possible, and if so, what can be the possible risk of this? Because nowadays risk related to carbon sequestration also. Yes, the similar way, if the N2O concentration, one thing, when I started my research in 2011, the average global value of N2O was 305. When I finished my research work, it was 320. Now, after 5 years, it is 330. So I am really against the climate deniers. I strongly support there is a climate change is happening. We are gonna face a lot of risk in the coming future. I hope I'm sure, like you all are aware about the changes in the atmosphere. I mean, the climate in the last few years, all over India. Not only in India. When I'm here in Middle East, here people are saying same thing. When I was in New Zealand, the people who are 50 years or 60 years old in New Zealand are telling always, this year it's very cold than normal, or this year it's hot more than normal. This is a truth. It's not like you can deny with many stories. This happened maybe also like 1,000 years back. But right now, in the last 250 years, we are facing a very serious threat to the atmosphere because of our intervention. It's very clear. Look at that figure. In 275 years back, the concentration of carbon dioxide was not changing. Why it is changing and increasing every year? We have some role. So we are obviously, to your answer to your question, yes, we are facing a big threat, not only in terms of N2O, in terms of carbon dioxide, methane, or there may be, a little bit, because we are aware now, we might be reducing, but still it's not under our control. It's going ahead. Hopefully, in the coming future, we all will be educated, and hopefully we will be able to So that's what I told you. To the answer to Muthuraj's question, we consider this as a mean value of the global air, or in our troposphere. That was 270 before 250 years. But after 250 years, in recent last 200 years, it keep increasing from 270 nanomoles until now. As I told you, in last 10 years, in my own research experience, it is increased by 15 nanomoles. So it's a very, maybe until 1950s, we had a pace. The pace now in the increase is a little bit more. No, that's different story. This N2O, which life science people were from medical science, is entirely different. We use N2O as a chemical for for anesthesia kind of things. Obviously, if it is in the ocean, I told you it's in nanomoles. If the concentration keep increasing and reach into micromoles or like that, organisms will die for sure. We cannot live here in the earth if it is reaching into micromoles, but hopefully it will not. That's all.
Yeah, I think you've answered most of them, or I should say you have answered all of the questions that our participants had. I would again ask if there's any other question from the participants. If not, then I would be happy to thank Dr. Nayeem Mullungal for coming for joining us for this FDP program. So your talk was really enlightened us with a very new and a possibly big concern which the world is going to face. So it was good to learn from a chemistry guy that what are the concerns about the N2O. I thank you once again for being with us.
Thank you so much for having me here too. And I thank Dr. SREE, and all of the organizers, the collaborating institutions, having a nice platform like this in this kind of world where we cannot meet together in a hall. It's a very interesting thing to talk and learn. So it was learning for me more than talking to you. Thank you so much.
Thank you, Dr. Nayeem.
All right. Bye.